And what I want to do today, because I rarely get this long to talk, which is great. They all seem to be 10-minute talks, my ones. So if I ramble on, do throw things at me. But it's a good chance, really, for me to talk about all the things that I think are interesting that we're doing, some of the things we're doing, some of the things in the future, and a lot of it will be unpublished data. But as Julian said, when I started in this area about 8 or 9 years ago, I guess, was the first sort of publications, everyone said this is a trend that's going to fade away, and we're still here, and if it is a fad, it's a growing fad, which is fine. Now, my disclaimers, and I'm an omnivore, and I do drink red wine, which will become evident later. And as some of you know, for the last 25 years, I set up the UK Twins Register. It has 13,000 twins in it, and is the largest in the UK. It's not the largest in the world, but it's the most detailed. So these people are probably the most well-studied people on the planet, with whole, initially with whole genome scans, and more recently, full phenotyping, metabolomics, proteomics, microbiome, etc. And we're still seeing them all. And the initial 10 years of my career was really looking at nature v nurture, where you compare identical twins and non-identical twins, work out that virtually everything you studied had a genetic component. The answer to everything is about 60% on average. Okay, so if you ever get asked that question, it's usually a good answer. There were very few things that have zero heritability. So those are the early days. Then we started finding genes and looking at SNPs for common diseases. But the last 10 years, I got more interested in why twins were sometimes not as similar as they might be. Why did actually age of death differ so widely between twins? Why did they get concordance rates for heart disease were less than 50%? Why, if one twin got breast cancer, the other identical twin only had about a 1 in 5 chance of also getting it? So clearly, there are other factors involved. And when you start looking at the age-related diseases which we're suffering from now, we also see that genes only play a relatively small part in the trends, the trajectories of aging, how rapidly people age. And so clearly what we thought was a nice slam dunk, everything's genetic, that's simple, we can all go home now, is turning out to be a little bit more complicated. And this obviously got me interested in what might be these other factors. And I did look initially at epigenetics, and I studied that in quite a lot of detail for about 5 years, and realized that yes, it played a role, but it was probably a rather small role, and it also was rather hard to translate that into clinical application. And the techniques really weren't there at the time, whereas the whole other area we're going to talk about, the microbes could be important. And the other thing that I started to see about why microbes might be important was when you started to look at some of the nutritional and dietary advice out there. And we're in a huge state of confusion, if you haven't noticed, about what is a good diet and what is a bad diet. And we have national guidelines that have been around for about 30 or 40 years telling us exactly how much fat, protein, carbohydrates, and calories to eat without any real evidence behind it, and on a basic assumption that everybody is completely average. If you look at this, one of the better randomized controlled trials of recent year, the DietFit study, it tried to answer the question, is it good to go on a calorie restriction diet that is low in fat or that's low in carbs? And obviously, if you're low in carbs, you're high in fat, and vice versa. And the answer was, after 12 months, it's a draw. These were 609 individuals from California who were overweight, and on average, they lost about 6 kilograms. So everyone said, oh, that's a waste of time. You can't tell anything from that. Well, what was really interesting is you dig down into the individual data, you can see that some people lost 25 kilos. This is the same diet. and others actually gained 10 kilos. So you think about those individual differences. What is it about those individuals that made them a really bad fit for that particular diet? And you starts to think that really this whole idea of there is one standard food that's good for everybody a bit crazy. Now, as the last decade has unraveled, we're really understanding more and more about the role of the gut microbiome. And I think we have to start seeing it now as an organ. We need to start talking about it as a virtual organ, the same way we would discuss the functions of the liver or the spleen or the pancreas or even the brain, because it interacts, it reacts to us, and also it causes those reactions. So there's two-way interactions in so many areas. And if you just look at these diagrams, they're getting more and more complicated every time anyone starts to draw them, because we understand they are actually doing more, they are interacting with more and more. And this, what we used to just call something like leaky gut, we now understand is related to subtleties in changes in pH, just by a small change in a few species of microbes. We're seeing that just some of the metabolites that we can measure well, like short-chain fatty acids, have dramatic effects. And of course, there's many others that we can't actually measure at the moment accurately. So we're only guessing about what's really going on. But we do know that these two-way interactions are working a lot. And only by discovering that, looking at these in more detail, we're really working out particularly what's going on in the brain, because initially there was no real idea that these had an effect on the brain because no one could think of any mechanisms. But every year there's another mechanism coming along about how potentially these microbes are interacting with our brain. And there's highly likely there are mechanisms we have no comprehension about how a signal, say, of getting some food in your mouth can, within seconds, via microbes, be transmitted to your brain and tell you how to respond. Now, this slide sums up probably the last 10 years of microbiome research in a very simplistic way, because if you look for the sort of similarities and the disagreements in microbiome research, I think you can see that there are very few cases where people have identified an individual microbe that is actually related to a disease. And so a lot of the naysayers of the microbiome said, oh, this shows, if you take the analogy with genetics, that they really haven't got a clue. It's just like genetics was in the 1990s before we got proper chips. You couldn't reproduce anything. But what we are able to do is reproduce the fact that there is something definitely going wrong with the gut microbiome, and the way we measure that is gut diversity, the number of different species. So this is extremely consistent from study to study, and there's an increasing list, so that every common chronic disease that we see has a loss of gut diversity. Some of this is coming as a result of the disease, but we suspect that a lot of it is also on that causal pathway as well. And it's disentangling that is proving tricky. Now, unlike genes, you can modify this, and your gut diversity, which is probably a better way of assessing anyone's health than a whole genome screen. So I can tell more about any of you from your gut microbiome snapshot than I can from a look at your gene sequences. We need to start understanding how we can improve the whole baseline of our gut diversity. Because if you extrapolated from this data and said, how can we get the whole populations to have a more diverse gut microbiome, that's what we need to be looking at. And everybody might be different, but there are some generic similarities. So a study we did with Rob Knight's lab, looking at the British Gut Citizen Science Project combined with the American Gut Project, looked at 11,000 people and found that the one common denominator about having a high, healthy gut diversity was eating 30 different types of plants every week. And some of you say, well, that's a lot. But actually, if you have lots of nuts and seeds and herbs in your food, actually a rather interesting sort of Mediterranean-style, Middle Eastern-style diet, it's pretty easy to get there. And if you live in a trendy area like I do in North London, most of the restaurants you go to pay a fortune for, those little plates, you can get that in one meal. So the idea is that basically these microbes are chemical factories, and therefore the more you've got of them, the more different products your body's going to make, the more you're able to cope with the stresses and strains of life and fighting off disease, etc. Now, we're also recently discovering that virtually all of the medications that have been studied in detail have some effect on the gut microbiome. And again, this is a two-way process. The medications may be altering the gut microbiome, like in the case of PPIs, where they change rates of streptococcus and therefore will slightly alter the acidity of the gut, which has some good effects if you want to get rid of ulcers and some bad effects if you want to have more infections. And increasingly, this list is getting larger and larger as we see the importance of the gut microbiome in explaining how people respond to different medications. Explains why people need different doses of opiates, why paracetamol works for some people and is completely useless for others, why antidepressants seem to work in only a minority of people, and in some people they don't work very well at all. And probably the biggest effect at the moment is on anti-cancer immunotherapies, where your gut diversity at the beginning of your treatment is the #1 predictor of whether you will survive 12 months later. This is things like metastatic melanoma. So these are huge effects, meaning that there must be really important interactions, particularly on the immune-type drugs, between your gut microbes and the effectiveness of those drugs. And just as an aside, mention that we've started collecting data. We're coordinating what's called the Prim Study, which is a multi-center study of melanoma patients undergoing immunotherapy, looking at their diet and their microbiome at baseline, as well as their immune status and metabolomics as they go through that 12-month process. And if anyone wants to join, please let me know. The biggest study so far has been around 20 or 30 patients published, and they've all been very significant. But clearly, to get that individual level of prediction about what a cancer patient should be eating or not, we do need larger numbers. But so far, everything is looking like we should be able to get some very big results out of this. And other people are also studying this in other cancers, such as kidney and lung. And so this understanding, everybody who's interested in the microbiome is going to have a role in the future, I think, in trying to work with oncologists in cancer therapies, especially as they all cost at least £100,000 per treatment. I'm sure you all know that microbes need fiber to produce many of their good products, and we tend to focus on the short-chain fatty acids, although there are hundreds of other products that they do make. And we know these are crucial for interacting with our immune systems, which line the gut, and that if you don't have fiber, studies have clearly shown that other microbes take over when there's nothing to eat, and they start eating your own gut lining. And this is the mucus layer of our guts. And once they get beyond a certain state, they start to cause problems. At a small level, it's all dose-related. At a small level, it seems to be very helpful, because all of us go through this every night when we're fasting. Unless we're one of these people who have to get up and go to the fridge every 2 hours. Generally, microbes like Akkermansia will be feeding off your gut lining, and that seems to tidy it all up and is what we need. You overdo the cleaning for too long and things can go wrong. And some of you might know this example, but when experimenting for my book, I was doing all kinds of strange diets. And I just finished the French cheese diet, where I was seeing what effect it would have on my microbes by eating only unpasteurized raw milk French cheese for 3 days, washed down with a glass of red wine. And I can tell you it's really fantastic for 24 hours, really energizing. By day 2, not quite so good, and by day 3, you really don't want to look at cheese again. And the next experiment I was going to do for the book was 10 days eating only at McDonald's. I wasn't very keen on eating McDonald's, but I wanted to see the effect on my gut microbes. Luckily, another volunteer turned up who was a hard-up student, actually liked McDonald's, and he also happened to be my son. So he ticked all the boxes, and essentially Tom did this study. But after 4 days, he came back to me and said, this is a lot harder than I thought. I really feel very ill. My brain doesn't feel right. I think I need to pack it in. And as a concerned father, of course, I said, no way. We're going to publish this in the Sunday Times. And that's exactly what we did. But the follow-up story is that he lost about 40% of his microbiome diversity in that time. And it took about 3 years to get him back to anywhere near where he was afterwards. So it's an N-of-1 study, but I think it does show that if you are depriving people of fiber for very long periods of time, it's very hard to recover the microbes that can then feed off the fruits and vegetables you might then be giving them in order to sustain a healthy microbiome. So we need to look into more about this relationship about fasting and lack of fiber, because they do go hand in hand, because you can be eating junk food that's all absorbed very high up in the intestine and effectively starving your gut microbes at the same time. So much more needs to be done to separate out these factors, but the fact that junk food might be working in ways we hadn't thought about, it's nothing to do with the fats and actually the sugars that could be interfering with our gut microbes in other ways. Now, we all know that we all have unique gut microbiomes. So everybody in this room has some unique species to them. And on average, if you look at the sort of around a sort of family level, we share about 30% or so of our gut microbes with each other, compared to 99.5% of our DNA. So we're pretty individual. And this has been one of the sort of problems about studying the microbiome is, if we're all so different, we've all got completely unique microbiomes, how do we make any sense of this? Because there's just too much individual variability to say anything. So, and this is becoming increasingly apparent as we've moved to metagenomes, because we get down to strain level and we see even more variability. But we looked at this by combining, in this recently published paper, a postdoc in our group, Alessia Visconti, by combining metabolomics with metagenomes in around 1,000 of our twins. And we looked at the metabolomics in the blood and in the gut itself. And what we see is that, yes, species, around 50% are really only less than in 1% of people. There's a few in common, but there's more differences than similarities. But if we look at the actual pathways, we see that there's much more similarity in pathways between people, so that our microbes might be different, but they're actually contributing to very similar pathways. So this might be an approach how we get around this individual variability by actually focusing much more on how do we address these metabolic pathways rather than these worrying too much about these unique strains and species. And what was also interesting, that there are only about 12% of our microbe species that were doing most of the work. These guys were doing all the crosstalk and were associated with the majority of the metabolites we could find in the feces. And just those 12% were also explaining around 15% of our circulating blood metabolites. So you could start focusing, forgetting the ones you don't know the function of and just focusing down on those that are the key players here. And this could be a really important role for therapeutics. I want to revisit where we are. So back in 2014, Ruth Lay's lab in Cornell and ours got together to do the first large twin study in the area of the microbiome, and also came up with a few candidate microbes that were protective against obesity. And the two big ones that came out were Christensenella and Akkermansia, and these have been replicated many times. Again, underlining this rule, there isn't a microbe causing something, it's usually an absence of a microbe causing it. They're generally protective, and so we need to sort of reverse our normal way of thinking here. But various companies have gone on to try and use these microbes to sprinkle it on your cornflakes or your muesli to make you thinner, but have had a lot of problems about getting these particular strains to either grow or actually be something that's actually therapeutically useful inside your body. And not everyone has large levels, particularly of Christensenella, although everyone has some form of Akkermansia. So we want to look a bit further, because this data was all in the days of 16S rather than metagenomes. We just need to realize that once you go to different levels and we start digging down, you can find different things, because the Christensenella result was also interesting because in that data it was about the most genetic heritable of the microbes we saw. But if we revisit these results, and you can see the old results in red here, and the newer ones in blue, where we've got over 1,000 twin pairs now, so 2.5 times the size, we can see that there is some heritability of all these microbes, and the more numbers you have, and once you move to metagenomes, you start seeing more genetic effects as your precision gets better. But still, the maximum around it is around 40% heritability, and some of these other ones drop. So the key was, are these important? At the time, we thought the ones that were heritable seemed to be the ones that we were finding were more related to diseases, which sort of made sense. You say, well, there must be a reason they're there. What's my reason they attracted one person and not another? Must like to be heritable. But we might have been wrong. And we've now, using metagenomes, opened up this whole idea of the Christensenella microbe into these subspecies. And we can see there's a big difference between them in their heritabilities, ranging from 0 to 68%. So these are very big differences that we've assumed was all the same. And so within this, we thought, oh, well, the Marseille version, that should be interesting to look at. But actually, when you look at that and see, is that the one that was causing the BMI change? It was to some extent, but it wasn't related to the visceral fat, which we think is really important. And it turns out that the least heritable one, Phil 17, is actually the one that's driving most of the obesity effects. So we're getting very different results here, and similarly explaining the metabolites you see in the intestine. So we'll be able to pick away at these by deeper metagenomes and increasing these studies. And again, the twin studies are still useful for this. Everyone knows these are all nice, good, healthy foods for our microbiome, and well, I should say our health. And it's turning out that we are understanding that why they are good for our microbiome. Now, obviously the fiber, the inulins, they're obvious. We're discovering more about the polyphenols as well. So it turns out that a lot of these foods which have flip-flopped about being deadly or healthy are now coming around to the idea they're generally healthy if they contain large amounts of polyphenols or fermented foods. So we're talking about the non-ferment or regularly non-fermented or those with extra polyphenols. And the one I particularly like is red wine, as I declared my interest earlier on. And we recently published this because it's been unclear the effect of alcohol on the microbiome. It's been rather hard to study, partly because people haven't taken very good histories. People lie, and there's lots of cultural differences in the way people use alcohol. But we took nearly 1,000 of our twins, where we had reasonable alcohol data at multiple time points, and then replicated in the American Gut Project around 1,000 were selected where they had actually good alcohol data, and similar numbers in the Flemish Gut Project. And we found that whereas total alcohol amount was not associated with diversity, red wine was. And we got a clear dose effect. We're able to replicate it in the other two populations. And we even found a few microbes that actually seem to go along with this. And we looked also at spirits and found nothing. There was a suggestion of effect for white wine. And that was quite interesting because in a way that's like a positive control socially, because there aren't really big differences between white wine drinkers and red wine drinkers apart from perhaps gender. But even when you adjusted for that, you still saw some weak effects of white wine. And the only real difference between the two is one has 3 times the polyphenol content of the other. So I think this is interesting because it's suggesting that we need much more work on polyphenols and polyphenol scores in foods, but also it's a good example of how you need to see beyond the nutrient and actually look at the whole food. Look at the skin, look at what actually is on it. It's not about the macronutrients, it's not about the back of the label stuff. We're doing similar work with yogurt. There's been a controversy about whether yogurt actually stays in you or gets flushed straight down the toilet and does nothing. We do see in our population of twins an association with alpha diversity when we've adjusted for all the obvious confounders. With 16S, we see an association with this abundance of streptococcus, but we can't see anything for bifido. It's rather strange. And when we use metagenomics, we do see the bifido coming out. And again, this is an example of how sometimes this extra precision does help you. And we see these consistent patterns here, but also we're also able to separate out 2 of the bifido strains. One was a commensal and one's actually in the yogurt, and actually divide that up. So again, going from the simple study to the more detailed one is useful. Just something that we're just preparing now is actually looking at the urinary microbiome, just to tell you that many people have bits of frozen urine in freezers, and the myth was that they were completely useless for microbes because they'd been so old and degraded you couldn't get anything out of it. But working with Rob Knight's lab, we've done a few thousand of these, and we are showing reliable, replicatable results. And what's interesting is that the diversity is going in the opposite direction to the gut. So a healthy urinary microbiome is actually less diverse. So it's looking more like the vagina than it is the gut. So we'll need to have a new way of looking at the urinary microbiome in that, but there are some clear associations with what's going on in the gut as well that we need to think about in the future. Now, coming back to my favorite topic of food, what if we all respond differently to the same foods? Now, the Weitzman group in 2015 had a paper on this, basically saying that microbes could explain your response to glucose better than pretty much anything else. The devil's in the detail, but certainly that was an exciting result that no one could actually explain at the time. And since then, I've been experimenting myself with a glucose monitor, which are now routinely available, and you can wear them for 2 weeks. They're affordable, and they give you a glucose readout every 5 minutes without any pain or suffering. And it turns out that if I drink orange juice, I get a much bigger peak than I do with Coca-Cola. And my favorite fruit, grapes, is the worst thing possible for me. And my go-to lunch of a tuna and brown bread sandwich in the hospital for 10 years turns out to be absolutely dreadful and puts me in the diabetic range. So you can learn a lot about yourself. What you thought was healthy turns out not to be. And to do this more formally, what we'd like to do is actually come up with a personalized way of eating based on some of these parameters that are all unique to us. Given our slightly different genetics, our extremely different microbiomes, many other factors, how can we get an app that would tell you how you would rank whether it's best to eat muesli, porridge, or brown toast in the morning? So to do this, we got together with a company who raised the money for this called Zoe, and the idea was to do a very large intervention study on twins, looking at the variability between people, what explains these differences, and can you actually use machine learning to predict this in some meaningful way. And the idea was to build on the idea from the ZV study, but make it more holistic. So include fats, include insulin, not just focus just on the glycemic response. We also added in the extra rigor of having a whole day in clinic. So we had 1,000 people in the main cohort and 100 people in the Boston cohort, all doing exactly the same protocol with these 10 blood draws, metabolic challenge meals, identical meals, and looking at all these other omics and questionnaires, etc. And it turned out that that was really crucial to get that baseline, because then when they went home, then they were a bit out of your control. And although most of them did what you said, it was quite hard to exactly control them like you would lab animals. But they did pretty well, really good compliance in all these testings. They were given digital devices as well as the CGMs to take at home. They also had blood prick testing to look at lipids after meals, sleep monitors, exercise monitors, and microbiome done at 2 time points for metagenomes and metabolomics. And they ate a lot of muffins. And can you guess why they're blue? That was to do with their transit time. So you can see this bright fluorescent blue color in the toilet because all the other studies have not really looked at this, but it turns out that actually transit time is one of the biggest factors determining everyone's difference in their microbiome. So it's a really important confounder. And we had over 2 million glucose readings and 28,000 fat readings. Only about 2.5% dropped out because they loved this working with the nutritionists and the app. Now, the main thing we showed is even bigger than I expected. It was an 8-fold difference between individuals in their response to the same food. Remember, these are pretty unselected normal people and huge ranges. So just showing that whether it's for fats, for sugars, for insulin, we all behave very differently with the same food. And this was reproducible, so it wasn't just error. And these are the individual variability was lower than into individual one. Now, what about its genetic effects? Because I'm a geneticist, so I've been trained to say everything's genetic. And certainly height is very genetic. If you look at the graph there, that's what you see when you compare identical twins for height. No doubt that you can predict one twin's height if you know what the other one is. You've only met one of them. If you do the same for your glucose response to a standard meal, you can see you're doing pretty badly. And you get about 30% if you knew everything genetic about that person, every single genetic marker, i.e. Having their clone. And for response to fat, it's even worse. It's somewhere between 0 and 5%. So in our metabolic responses, genes are really not a major factor at all. And as you know, there are lots of companies selling home kits to tell you which foods you should eat and how you respond based on DNA. Even identical twins had different responses to food. And this was a surprise. We thought they'd be matched for things we didn't know about, and that would be important. But going back to our other findings that most identical twins often have very different microbiomes, it starts to make more sense. We broke all this down with this arm machine learning to say, well, how much of this response between all of us is related to our individual responses to food? And I think you can see that we explained more or less around 50% of the variability, and most of it was individual characteristics rather than the sort of things you read on the back of a packet. So rather than your macronutrients, or you're saying how much fat or how much sugar, it played some role, but most of it was down to the individual and the individual's interaction with that food. In terms of the microbiome, we got a smaller effect of the microbiome of around 5 to 7% for sugar, and it was greater for the fat levels. So in how you respond to fats, presumably through the bile salts and all these other, at least some of the known mechanisms, it sort of makes more sense that your response to how quickly you'll clear a fatty meal is to a large extent as a modifiable factor determined by your microbiome. And I think this is a really interesting thing to realize here. And that actually got, if you separate out really young, healthy people with no disease at all, that got even greater. And so all this idea about going on low-fat diets or high-fat diets could depend on the microbes that you've got that are, in a way, able to deal with a fatty load either worse or better than other ones. Because we believe that if you've still got lots of fat particles floating around your body at 6 or 7 hours, you haven't cleared them, these are the things that actually cause inflammation to the arteries and actually will make you gain more weight and more fat will go into the adipocyte. Obviously, there are other factors in here, such as circadian rhythms, whether you're a morning person or evening person, whether you should skip breakfast or skip your evening meal. These come down to very individualistic differences, which we're still working on. Obviously, machine learning is one of the other big technologies that's come along in the last few years that, combined with the technology, this supercomputing method of putting things together, once you've got large data sets with thousands of people, hundreds of thousands, millions, you can make very good predictions. And already we're up to about 77% accuracy in these predictions. And this validated in the US set. So although clearly there are differences between the gut microbes and the habits of Britons and Americans, within a trial design of similar people, you can get similar results, which is reassuring. So we don't have to do this for every micro population around, which would be pretty impossible. So the conclusion of that study really is everyone's unique in their food response, even identical twins. Genetics explain less than a third of the response, and the composition of the meal is not that important. And there are ongoing studies to do this in the US, getting thousands more people through this beta testing of this app that will help our models. So our future plans, we've started, and we should be finished within about 6 months of getting 4,000 metagenomes on the twins. We're going to combine this with the metabolomics using mass spec predominantly, which in the gut seems to be the most useful one. So you can identify at least 800 different chemical metabolites reliably in the gut, and then start to combine that with what's going on in the blood. Because I think this interaction between just picking one and not knowing the other is somewhat difficult, because if you see high levels of a metabolite in the gut, you're not quite sure whether the microbes are producing it or your body's eliminating it. So sometimes you need a ratio to be sure of that. The urine is looking increasingly useful, particularly for studies of aging. And we're also collecting 200 twins where we've pretty much got everything on them. So they've had colonoscopies, and we've taken samples from their saliva, their urine, their gut. We're looking at the ileum. We're getting cecal fluid, which is presuming very useful for metabolomics as well, and combining that with our genomic idea. And of course, with all these food responses, we're also finding some microbes that are associated with them as well. All this, what's going to help personalize our nutrition. So I want to acknowledge the huge numbers of people that make this happen. Just in the PREDICT study, there's about 100 people involved in that study, which is still ongoing. And within my group, there's probably about 20 people, but the key ones here, the two postdocs, Caroline Leroy and Alessia Visconti, who've done the sort of hard graft of the new data I've been showing. And really to sum up, I think the best way to think of your gut microbes is in some ecological context. And so I like the garden analogy. And I think we've got to realize we've all got unique gardens inside us that we need to nurture. And the microbes are key to this uniqueness. And each of our own unique microbes has a unique interaction with the foods we eat. We have to realize that. But importantly, so it's not too out there, we do share a lot of metabolic functions in common. And this, I think, is very much the way forward in trying to unravel this, that there might be different ways of getting that function to work. And combining metagenomes with metabolomics is helping us to unravel these key functions. And realizing the potential for personalized diets, I think, is going to be very important. I might explain also why we've got so many anomalies in a lot of the trials that go on in the microbiome, which are just too small, and they aren't realizing the different starting points everyone's at. So I think we've done a great job so far knowing these problems and these variabilities between the studies that we've got. But lastly, we need to encourage diversity for our own microbes, and to do that, we need to encourage diversity of everything we eat, particularly plants. And remember, with 100 trillion microbes inside you, you'll never eat alone again. Thank you.
Thank you, Tim. Thank you for a very stimulating talk. And so I open the floor to some questions.
That was fab, Tim. Thank you. Quick question. Have you come across the work of the Israeli research group with Neutrino, the app that they were developing to try to personalize this data?
Yes, well, I mentioned it. That came out of the ZV study.
Yeah.
So, and they had a spin-off company called Day 2, which is based now in San Francisco.
Oh, so that was the evolution of that research?
Yes, so it spun off with a Weizmann, but they moved to the US, as most companies do. That's where the money is. And they are, as far as I know, I'm not involved with them in any way, but as far as I know, they're focusing primarily on pre-diabetes and diabetes, and trying to get this app that diabetics can use to try and improve their glycemic control. But they're focused purely on the glucose side of it, whereas what we're trying to do is slightly more holistic, and also, because it's quite easy, I can tell everyone how to control their glucose, it's just eat fat and add ice cream to everything, which is not a bad way to do it anyway, but it's...
And a glass of red wine now and then.
Yes, exactly.
Thank you. Thanks.
Any more questions?
Thanks for the talk. I'd like to ask your view on the generalizability of this relationship between diversity and a healthy microbiome. And a lot of the data you presented is from Western populations for sort of chronic health conditions such as diabetes and metabolic disorders. If you take, say, the developing world and young infants in that setting, we might be seeing a different picture where the health issues of sort of infectious disease and stunting. Is the same relationship of diversity and microbiome health going to hold in different ages and across different geographic populations?
Right. Very good question. Looking at the work of people like Jeff Gordon and some of the data that's come out of Bangladesh, etc., about these young kids, and you have to realize that what I was talking about really was adults. That's what I focused on. That's was before the age of 4, things are very unstable. So these general rules don't seem to apply, and you get very large fluctuations in the gut microbiome. It just doesn't settle down until about the age of 4. But at the age of 4, then there's not much difference between a 7-year-old and a 50-year-old, interestingly, in terms of if you take a very broad brush view, which is very unusual in the rest of medicine or physiology. So I think those people are really focusing on if you can really maximize what happens in particularly the first 3 years of life, then you can set the course, get normal growth, etc. If it's very impaired in those first 3 years, then you may never recover it totally. So I was referring really to case control type studies of adult diseases, predominantly in the West. But when there is a good association, like the Christensen-Eller association with thinness, for example, or negatively associated with obesity, that has been replicated in at least 4 populations, including at least 2 in Asia. So for some conditions, there do seem to be these associations. For others, where there are microbes that seem to have different functions in different countries. So Prevotella is proving particularly difficult because it seems to have different effects. It seems to be quite healthy in Africa and very unhealthy and inflammatory in the West, and there's other examples like that, where microbe might be context dependent. But I think I was just trying to give areas where there is sort of consistency. There's plenty of areas where you can pick it apart, and I didn't mean to summarize that. Actually, it's very easy. And once you go down into the detail, as we're doing, even down to strain level, you'll see all kinds of inconsistencies because of the complexity and the fact we don't know exactly which microbe is in which community and what it's doing. So it could be that a Prevotella in Africa is working with other microbes that are beneficial, and it's in a different network in the West, and therefore it's having the opposite effect. So, but thank you for pointing that out. It's very important that there's a clear difference between the first years of life and afterwards.
Hi, Tim. So I was interested, it sounded like when you were talking about with the personalized nutrition, that it sounded like you were much more interested in the triacylglycerol response. And I was wondering if, because it seems, of course, this is not my area of research, but that with personalized nutrition, so much in the microbiome, there's so much emphasis on the postprandial glucose response. So I was wondering if you do believe that the triacylglycerol response is much more important, and if so, why that's maybe not being investigated as much as the glucose metabolism.
Okay, so we found that the postprandial triglyceride was actually more predictive of cardiovascular risk scores than just the fasting one. And you've got to realize that we spend most of our life in a postprandial state, not a fasting state. So the reason that modern medicine has just come down to this, you just take this in this one narrow window when everybody is actually very similar, is a fallacy. We should really have, I can understand why it happened, because it was a way of labs particularly standardizing. They like low coefficients of variation. They want nice narrow areas so they can give a nice normal range. But actually, when you're trying to predict things, you want the opposite. And so what's clearly showing is you can have a perfect fasting triglyceride, but you're way out there on the top of the scale once you've had your pound of cheese or burger. So that's the historical reason that it's been there. Also, it's hard to measure. You think about having to take a standard meal and measure 6 hours later your blood. The reason it hasn't been done is there haven't been ways of measuring it without staying in hospital, without having a standard fatty load. And so there's lots of reasons. And also we have a glucose monitor now that is incredibly cheap and easy to use. We don't yet have a continuous lipid monitor, although I'd love someone to invent one. So we still have to prick your fingers. They're the main reasons. But I think I can see the future moving very much towards that. I don't see we can stay with this fasting level being the gold standard anymore, particularly with all these latest results.
Thanks, a great talk, Tim. Now, being a dermatologist by upbringing, I noticed that you didn't talk about the skin microbiome. I think you have done some work around skin as well. I just wondered whether there is a story in terms of our diet, nutrition, and skin health, and not necessarily the role of the skin microbiome in the context of skin disease like atopic eczema or psoriasis, but in a healthy state, are there differences depending on what people eat? Is that something that you've looked at?
Yes, well, every part of the skin has a different community. Everyone is unique in their skin just as they are in their gut. And there's increasing evidence that most of the common diseases are related in the same way to microbial shifts or differences in the microbiome for skin disease. So eczema, psoriasis, allergies, acne, all you can see changes, and antibiotics will have some effects, might be short-term or long-term, on these skin conditions. So there's no doubt that the same thing is happening. It's just very much harder to study it because the technology of getting metabolites from the skin is harder. We've tried it. It's possible, but it's really hard to use sebutape and desquame to get accurate idea of what the chemicals are the microbes are producing rather than something else. So I think it will come. It's just going to be a bit slower than the rest of the field. But there are definite links between what's going on in the gut microbes and skin diseases. There's absolutely no doubt of that. So they are all related. Again, we just have to wait for the technology to catch up because they're low abundance microbes. And once you start getting low concentrations, contamination is very easy. We haven't really got the best method yet of swabbing people on very large scales to get that kind of data. Does that answer some of your questions at least?
Fergus.
Tim, I've no doubt that food diversity and microbial diversity are a good thing in people who have a low food diversity and a low microbial diversity. But most things in life that we think are good for us, including red wine, they're good for us up to a certain level, and beyond which they stop being good for us, and maybe even we get to another level where they're actually harmful. So do you have any doubt that maybe we're overstating some of the diversity issue? I mean, it is self-evident and a no-brainer that, you know, in modern life, we've a diet that's fiber depleted, that's roughage depleted, that's diversity depleted in comparison with ancestral microbiomes. But have you any doubt that maybe we're overstating some of the diversity issue now?
I think there's two parts to that question. One is about fiber and one is about diversity. There's lots of recent studies showing that actually in randomized controlled trials, and we've done one, where you give people a very specific fiber like inulin, you actually can drive down the diversity of the gut microbes. And so in certain people, giving too much high levels of a very specific fiber could actually be bad. So I think this is quite a new concept, which in a way comes to this point that, yes, unlimited fiber and may not be good. And so we've got to get the balance between are the resident microbes that are there able to cope with what you're giving them. So you could possibly overload, if we all had 20 grams of inulin every day, it would probably overload our systems. And there are companies out there trying to sell those things to us. And I think we have to be very wary of the artificial type fibers that are highly concentrated in one particular area. But I don't really know anybody who's had a diverse plant diet containing lots of different fibers that has had that similar level of problem. Although at the meeting in Ireland, there was I can't remember exactly where it came from. I think it could have been an Irish study that just a general high fiber diet also had the same reduction in diversity. So if people are not used to high fiber diets and then they're given very high, you might see at least transiently a drop in diversity because they don't have the microbes to deal with it. In the same way my son maybe just didn't, they'd all died off. So you may need to think about how you repopulate some of those good microbes in order for the foods to work. So I think as we're doing this, we're understanding more, we're realizing there's always got to be a balance. There's no such thing as unlimited. It's like, yeah, exactly.
Well, I don't know.
Yeah, and it's the same idea with vitamins, We thought, oh, well, vitamins work in tiny amounts, so if you give masses, they're great for you, and it turns out they're not. And I think the same thing could end up being true here. We've got to get the balance right, but at the moment, most people are so deprived that that's not our major problem.
Hello, you mentioned the urine microbiome a couple of times, so I was wondering what are the safeguards you're using to prevent against contamination as you have contamination during delivery of the urine sample and it's a low biomass sample as well?
We use the same principles as collecting a clinical midstream urine. So you don't collect the first bit of the urine, you ask that bit's discarded and you collect the bit that's flowing. You can't control it perfectly when you're collecting thousands of samples, so it's quite likely there is contamination. And there's no way around that. So I think you have to accept there will be some contamination of all these samples, and that's possibly why in that whole perineal microbes will always come out into those samples. In terms of these low mass areas, which is a bit like doing the skin or the urinary biome, you just got to have a lab that's very careful and is used to doing it and isn't right next to your one that's doing the feces. So that you have to have separate labs to do that and just take a lot of care. And it's often trial and error. And that's why we didn't attempt it straight away, because we weren't ready to do this 3 years ago. We could probably do it now, but a big lab like Rob Knight's in the US was the place to go. But you're right, there are a lot of difficulties of doing it, and you need to know what you're doing and check you can do it reproducibly.
All right, we're running out of time here, and so I'm going to take the chair's prerogative and ask a question myself. Sorry, guys. So I can see you're an advocate for the metabolomics. What about the proteomics? Is that the other axis that we should be looking at in way the microbes are interacting with the host, especially in immunotherapy via the immunological axis? And what's your thoughts on the proteome of these organisms and the metaproteome?
It's definitely the next omic to start thinking about, particularly the interaction with the immunome. That's going to be crucial. At the moment, we do still lack the really good tools, and we've done some limited proteomics in our data. A lot of NMR is not useful enough to identify exactly what you're doing, although you can get lots of data. It's just lots of colourful peaks, which look good on graphs, but aren't actually very good for anything else. For our kind of work, anyway, I'm not saying they're useless, but and the other ones where you're doing a sort of mass spec type precision, it can cost you £1,000 a sample, and they're currently locked up in several companies that are doing this. But that's probably the way forward. Plus, we are doing a big study of 2,000 of our twins using a mass flow cytometry for all the immune cells. The problem is the guy doing it, Mario Roda in the NIH, he said, well, okay, Tim, I've got 1 million different subtypes for you to look at. And that causes a bit of a Bonferroni problem when you get to doing it. So I think, but it's something I definitely want to do. I just think we haven't quite got the right technology to look at it. It's certainly the immune proteins that I think will be the next big breakthroughs. And I think one way of doing it is to see these massive changes in these cancer patients, because, you know, if a quarter of them are surviving and a quarter of them are dying, and you can look and see which microbes were driving those immune changes, that could be the shortcut to this. And I think, I'm sure I'm not the only one looking in that space at the moment. But the other problem is you often have to collect PBMCs and things like this and actually plan the study a lot ahead of time. It's not something you can just go back to 10 years later, unfortunately, but definitely worth looking at.
Okay. So thank you. In the interest of time, I'd like to thank our speaker, Tim, again for a very stimulating talk. Thank you. Thanks, Tim. Thank you.